Thank you so much for being here. Thank you for spending your Saturday with us, for celebrating Citizen Science Month with us, Earth Day, the City Nature Challenge, all the great things that are going on in April. And especially in light of emerging public health concerns, I know I am personally really excited by these events, and they mean a lot to me. So thank you to you all. So NYBG, Charles, do you want to describe this?
Sure. So thanks again, Carolyn. My name is Charles Zimmerman. On the behalf of the New York Botanical Garden, I want to thank you all for joining us for our final day of our 4-day program celebrating the 50th anniversary of Earth Day. With me here is Carolyn Nickerson, who's already identified herself, and my colleagues at the New York Botanical Garden, 3 amazing individuals, Daniel Atha, Christine Paulus, and Esther Jackson. So during this first hour, each of us will share an introduction to the projects that we lead here at the New York Botanical Garden to engage the public in scientific discovery through exploring nature around us and safeguarding knowledge about plants, which we all know the survival of our living Earth depends. So in the second hour, you'll have a chance to do citizen science firsthand as we break out into groups, and the 4 presenters will lead interactive workshops on the projects we introduced in part 1. Each of you will be invited to join the group of your choice at the end of this meeting, and Carolyn will tell you a little bit more about how that works. As you learn today, it's never been easier or more important to get involved as a global citizen in projects to investigate nature and protect our Earth. So that said, public participation in science has really deep roots at NYBG. A great amateur naturalists like Job Bicknell Ellis were instrumental since the garden's founding. Starting as a teacher and a farmer with no botanical training, Ellis was inspired by curiosity and fascination with mushrooms and came to be one of the most prolific collectors of fungi of all time. His personal collection of over 100,000 mushrooms came to NYBG in our earliest years and are still used today by scientists around the whole world. So the issues we face today, from environmental degradation to our isolation from nature, not to mention the current global health crisis that we all find ourselves in, they're enormous and they're unprecedented for sure. Thankfully, we have incredible new tools for curious observers like you to discover nature, connect with one another, and support the conservation of our Earth no matter where we are. So at this point, I'll hand off the mic to Carolyn again so she can lead our ceremonies for the rest of the afternoon. And thank you again, everyone, for tuning in.
All right, so we're going to get things kicked off with a live poll. So I want to know if you have ever participated in a citizen science project before. So I'm going to launch this poll in just a second. All right, it's launched. So please vote. Yes, no, not sure. All answers are okay here. And if you don't know what citizen science is, that's okay too. You'll learn.
Can we participate?
Oh no, panelists can't, unfortunately. Your votes.
Oh yeah, I'm trying to click here. I'll put mine in the chat.
All right, we have a lot of yeses pouring in, a few no's, a few not sure's. I'm gonna end the poll in just a second. Someone said bias sample. Yes, this is indeed what we call a convenience sample. All right, 3, 2, 1, we're ending the poll and we're sharing results. So we have a majority for yes, a few no's, and a few not sure's. So we have one more question to kick us off. We want to know a little bit more about you. You know, if you are an aspiring citizen scientist, if you are a parent of an aspiring citizen scientist, you can select all that apply. So we're going to go ahead and launch that poll. So please feel free to vote. Let us know if you're an educator, a teacher, if you're a librarian or part of a library staff. If you're just a bored person on the internet, that's okay too. If you're none of those, that's fine as well. All right, our votes are pouring in. We got a few bored people on the internet. We got quite a few teachers and educators. That's great. And almost 50% aspiring citizen scientists. So we're going to end the poll in 3, 2, 1, end poll. And we'll share our results. So yeah, 50% aspiring citizen scientists, a few parents here, some teachers, some educators, 2 librarians are with us. Hello, welcome. A few bored people and some none of these. Well, by the end of today, you will all be citizen scientists. So you'll have a new title. So this is me. I wish I could be there with you in person, but unfortunately we're all online, but we're still able to connect and still able to do amazing work, which makes me really excited. So feel free to tag me on Twitter, get in touch with me via email. I'm always happy to be in touch as a member of the SciStarter team. So SciStarter partnered with the NYBG for this particular event as part of our Citizen Science Month initiative. So that runs all through April. And it basically means we want regular people doing real research every single day of April in whatever way they can. Citizen science, I always call it a collaboration. It's between scientists and those of us who are curious, concerned, and motivated to make a difference, which means that you get to be involved in real science, advancing knowledge, created knowledge. And someone just asked in the chat, will this be recorded? Yes, it will. It will be recorded. And for my email address, yep, it'll be available in the chat. Actually, right now, I'll go ahead and put it in there in case anyone wants to get in touch with me. And I'll be in the breakout sessions as well. We have some great partners for CitSci Month. We've worked with Science Friday, Nat Geo, we're doing a webinar with them next week. The National Library of Medicine have been awesome partners in selecting projects for libraries. ASU, the Citizen Science Association, we're just so grateful for everyone and every single citizen scientist who's planned an event. And if you want to share your excitement during this presentation, feel free to use the hashtag CitSciMonth. So for the sake of time, I just wanted to briefly describe a SciStarter account. We hope that you all, after you participate in the projects today, are willing to do even more citizen science. So please go to SciStarter, scistarter.org/login, and you can search different projects there. There are over 3,000 citizen science projects listed in citizen science events. And you can make an iNaturalist account. If you have a Zooniverse account, you can keep track of your participation in SciStarter by going to your SciStarter dashboard. So that's in your info and settings, and adding your Zooniverse and iNaturalist usernames. These are many Zooniverse and iNaturalist projects are affiliate projects, meaning you can keep track of the number and frequency of your contributions in your SciStarter dashboard. So after you do these awesome projects from the NYBG, there's so much to explore. And with the NYBG team here today, you'll be able to just dive right into citizen science. So that's for later. I'm going to go ahead and stop sharing my screen now and hand it over to our first panelist, which is Daniel.
Hi, everybody. Hi, my name is Daniel Aita, and I'm the Director of Conservation Outreach at the New York Botanical Garden. I'm so glad to be with all of you virtually anyway. I'm glad we can share this time anyway together, maybe not the same space, but thank you, Caroline and Charles, for your organizing this event, putting it together for all of us and giving us this platform to reach a broader audience. Appreciate that very much. And I'm going to present the New York City Ecoflora Project, which is a community science, citizen science project to document all of the wild plants that grow in New York City. And we're doing this project with all of the enthusiastic participation of all New Yorkers. And so it's really a community-wide effort to document all the plants. And I really look forward to sharing this project with you this morning or this afternoon. And then we'll, in the breakout session, you'll have an opportunity to actually participate in one of our research projects building on the observations that all of you have made over the last several years. We're leveraging these observations to maximize their scientific potential. So stick with us for the whole afternoon, I hope. You'll have other options for participating as well. So without further ado, I'm going to share my screen. And you should see the Ecoflora presentation. Is that right? Thumbs up if you can see the screen of the presentation. Okay, great. Now, just one second. Okay, technology under control here. Thank you for bearing with me. So the New York Botanical Garden is an advocate for the plants and fungi that produce oxygen, clean water, and hold the soil. They give flavor to our food and provide inspiration to our cultural lives. As a scientific collections-based institution with some of the most comprehensive botanical resources in the world, the New York Botanical Garden is uniquely capable of providing the community with science-based information on plants and their ecological dynamics, informing management and policy so our planet can be managed wisely and sustainably. The New York City Ecoflora combines the New York Botanical Garden's historical strengths in collections with emerging opportunities in digital technology and community science to maximize the impact on plant conservation in New York City. All New Yorkers are invited to participate as community scientists to observe, collect, and compile information about the city's plants and their relationships with other organisms such as birds, insects, and mushrooms. Superimposed on this beautiful, wonderful map from the National Geographic Society is a graph of the number of organisms present on Earth over the last 500 million years. And this is the total number of genera on Earth over 500 million years. And that's the yellow graph you can see moving up to the right. Immediately obvious is a lot of fluctuation and periods of rapid evolution and rapid extinction. These fluctuations can be grouped into 5 mass extinction events. You can see the extreme dips, where approximately 75% of species were wiped out in a relatively short period of several million years. Several million years being a relatively short period of time over 500 million. What is remarkable to me is following each extinction event, the diversity of life on Earth came back greater than it was before. You can see the trend line is always going up. The trend is always upward. The last dip on the right near Australia is the 5th mass extinction when the dinosaurs were killed off about 60 million years ago. The very tip of the line on the far upper right represents the last 10,000 years. This is a geologic period, epoch, called the Holocene, during which the Earth achieved its greatest diversity of marine and terrestrial life. You can see that it's 4 times greater than it had ever existed on Earth before, just until very recently. The Holocene was marked by a relatively mild climate across much of the globe, with a protective atmosphere of 78% nitrogen, 21% oxygen, and 0.04% carbon dioxide. And we all know that number is rapidly changing. Scientists tell us we are now in the Anthropocene, a new epoch, a geologic age so dominated by humans that our presence will be detectable in the rock strata millions of years from now. There is debate about its exact beginning. Some say it was 1850, others more recently. But it's safe to say that we are the only creatures in the 4 billion year history of life on Earth that has witnessed the transition from one epoch to the next. From the Holocene to the Anthropocene in our lifetimes. We set out to conquer nature and in the process have unleashed the 6th mass extinction. Ecosystems, species, and life-sustaining processes are being pushed to the brink of total collapse. Plants, animals, fungi, and untold other microorganisms are going extinct. All heard the numbers. 30% of birds are gone. Amphibians are on their way out. Corals are dying worldwide. 70% of insects in some places are completely wiped out. Alaska is on fire and the Amazon is drying up. Pandemic diseases are increasing. We are realizing that to conquer nature is to commit ecological suicide. Crucially, this realization comes at the nadir of our connection to nature, when fewer and fewer people recognize or know hardly anything about the plants, animals, and ecological processes around them. It has become so pronounced that the subject of this article in the Wall Street Journal, PhD agriculture students couldn't even recognize the plants they were studying in the laboratory. At the New York Botanical Garden, we've been conducting research on plants and fungi for 128 years, amassing the second largest collection of preserved botanical specimens in the world, educating generations of scientists and promoting plant conservation around the world. Building on the institutional strengths of the garden's library, herbarium, and scientific staff, the New York City Ecoflora is leveraging technologies like mobile computing, social media, comprehensive assessment of the native and non-native wild plants of New York City ever assembled. We are bringing big data to botany at a pace needed to keep up with the Anthropocene. Charles Darwin was driven by a sense of adventure and great curiosity, the same motivations that inspired the founders of the High Line to explore an abandoned railway in the meatpacking district of Lower Manhattan. The High Line is now one of the most popular parks in the entire world. Was one of the biggest tourist attractions in New York City. They will come back. Nearly everyone who visits the High Line says, wow, I wish I could go back in time and see this place before it became a park. Well, New York City is full of High Lines. They are in every borough, in every neighborhood, just waiting to be explored. In this image, we see Ecoflora volunteers observing ailanthus trees along Newtown Creek on the border between Brooklyn and Queens. On the day we visited, we found monarch butterfly caterpillars and the five-angled daughter, a New York State rare species. Field trips like this and other Ecoflora events bring people together to explore the city and the amazing diversity hidden in every neighborhood. They also provide an opportunity for us to learn from each other, share stories, and strengthen community bonds around our mutual love for nature. Working with professional researchers, community scientists have made important natural history discoveries. Suzanne Dickerson, a resident of Colorado Springs, trained a backyard webcam on a visiting skunk that was coming to her back porch every night and discovered the animal using a rock to break the ice in her dog's water bowl. She shared her findings with local researchers, and together they published a paper on the first example of tool use in skunks. An amazing natural history discovery made by a citizen scientist, the webcam in her backyard. Eve Levine, a resident on the Upper West Side of Manhattan, discovered a wild spurge plant near her mother's apartment and posted her observation to iNaturalist, a free, non-commercial biodiversity program. Her plant was identified by Nathan Taylor, an expert in the plant group of the spurge group 3,000 miles away. When I saw Eve's observation on iNaturalist and Nathan's identification, I realized the plant had never been seen in the wild anywhere in New York State. Within days, the three of us published a paper together documenting the plant's presence in New York State for the very first time. For those wishing to take their enthusiasm and curiosity to another level, the New York Botanical Gardens Adult Education Department offers courses throughout the year with in-depth instruction on the birds, insects, mammals, and ecological communities throughout the city. We are training a corps of super urban naturalists who will monitor the biodiversity in their communities and help their neighbors engage with nature. The Neighborhood Naturalist Program, pioneered by the New York City Eco-Flora Project, are neighborhood groups that meet regularly to discuss local biodiversity concerns. Because nature has no seat at the table, neighborhood naturalists act as the voice for nature in their communities. They also educate their neighbors and inform policymakers who rarely hear the plant or animal's perspective. Recognizing that most environmental degradation happens at the local level, neighborhood naturalists attend local board and council meetings and speak out when nature is threatened and when there are opportunities for positive change. To date, the Upper West Side Neighborhood Naturalist Group has promoted the use of bird-safe glass in new construction, the use of native plants in landscaping, and the recognition of light pollution as harmful to insects and other animals. Neighborhood naturalists are a regular presence at board meetings and faithfully advocate for nature at every opportunity. The New York City Ecoflora is the first of its kind real-time flora built by scientists and sustained by community participation. With our botanical garden partners across the country, you can see on this represented by the logos on the screen here. We are building the scientific literacy and community engagement needed to protect our wetlands, our forests, and our urban spaces that support life. It is a global model for documenting biodiversity and ecological dynamics to build a sustainable future and the inspiration and well-being of all. We are extremely grateful for the Institute of Museum and Library Services for their generous National Leadership Grant to the New York Botanical Garden and the New York City Ecoflora with making this project possible. And with that, shall I take questions now, Caroline, or shall we save them for later?
Let's save them for the breakout.
Okay.
So we can go ahead. Thank you so much, Daniel. I learned a lot from that presentation.
Great.
And I think next up is Christine. So Daniel, if you want to stop sharing your screen, we can let Christine share hers.
Yes, getting there. 1 second. Okay.
There we go. All right.
I'm getting better. Thank you.
Thank you so much. Christine, you're up.
Okay. Hi there. I'm Christine Paulus, and I work in the horticulture department with NYBG. And I'm gonna start my PowerPoint. So later on in my breakout section, I'm gonna show you how you can use an app called Nature's Notebook to record nature's calendar. Here's a little review for some of you. We're going to talk about what is citizen science, what is phenology, which some of you might not know, why should we collect phenology data, and I'll talk about how to collect data at home and how your data are used. Citizen science, also known as community science, is the participation of the public, people like you, to collect scientifically accurate data, and it's used by actual scientists. The phenology program at NYBG is just one of many citizen science programs at the garden. Phenology is the study of the timing of life cycle events in plants and animals. So it is the seasonal changes in plants and animals. We're going to focus on plants here in this session. It's nature's calendar. So it's when flowers are blooming, it's when plants are fruiting, when leaves change color in the fall, when they lose their leaves in the winter. Those are all phenophases, all part of the phenological cycle. Working with the National Phenology Network, NYBG engages volunteers to collect data on our living collections at the garden. Our volunteers are trained by staff, and they learn how to identify the plants and learn how to identify the phenophases. The data collected by our volunteers volunteers in our special events and public programming departments for events such as Daffodil Weekend and Lilac Weekend, so we can help predict when our plants are going to be in peak bloom. Our data are also available through the USA NPN database, the National Phenology Network database, and is used by scientists to study the impacts of climate change. And that data is available to anybody who wishes to access it. Data are entered in an app called Nature's Notebook. We also upload the data from Nature's Notebook into our collections management database. You can also, for those of you who don't have smartphones or tablets, you can also collect data. You can download and print a data sheet and carry it out into the field and collect data that way. We'll talk a little more about that at the breakout session. But most people use it on their phones, download the app, and we'll go through step by step how to download the app, how to get the app, and how to record phenophases. The data that we collect is also used in our living collections database, which is publicly accessible by anyone online. It's called Garden Navigator. You can search by plants, find out how many specimens we have, and see a map. You can also see the phenology data that is collected by our volunteers. And this is a screenshot of a page from Garden Navigator. This is a record for red maple. Here you can see where we list all of the specimens that are accessioned. These are specimens that are part of the living collections. And here's the phenology data. So you can see when flowering and fruiting phases have been observed and recorded. This is not to say that it's every single time red maples have flowered or fruited, but it's when we have recorded them and when we have entered them in the database. Our program is just a little over 10 years old, and every year we collect more and more data. Of course, right now our volunteers unfortunately are unable to collect data on the grounds at NYBG because we're temporarily closed. However, for those people who would like to collect data at home, you can set up your own site. You can set up a site in your backyard. You can even set up a site to monitor the trees on your street right outside your building. You can also go to a local park and add plants. So in the breakout session, we'll go over step by step how to use the app, how to set up a site, add plants that you see, and how to record that data. And once your data are recorded, that's available for anyone to use. And that's all done in the app Nature's Notebook or through the USA NPN website if you prefer to download paper data sheets and print them. We remind you to practice safe social distancing if you are doing it outside in a public space such as a park, and make sure that you are aware of your surroundings and keep a distance of 6 feet between yourself and others. So we're gonna walk you through setting up a National Phenology Network Nature's Notebook account. We're gonna create a site with the plants. We'll show you how to look for plants and add them to your site walk. And then I'm going to show you some resources where you can learn about this species, learn how to identify them, and also learn how to identify their phenophases. You'll learn how to identify what a leaf bud looks like or a flower bud, how to differentiate between unripe fruit and ripe fruit. And then we'll show you how to record those phenophases in your observations. So I hope to see you all at 3 o'clock in the phenology breakout session.
Awesome, thank you, Christine.
I'm gonna hand it over to Charlie. Oh,
that's right. You can go ahead and stop sharing your screen and then we'll pass on the mic.
Yep.
All right, there we go. Charles, are you ready?
I am ready. Thank you. Thank you, Christine, and thank you, Daniel, again, for sharing about the great projects that you do. Hold on, let me just share my screen. There it is. So my name is Charles Zimmerman. It's my privilege to, can everyone hear me and see me?
Yep, we can hear you and see your screen.
Great. It's my privilege to talk to you about another way to take action this Earth Day and beyond, and participate in citizen science from home through NYBG virtual herbarium expeditions. So by viewing photographs of and deciphering collection labels and recording information about the origin of wild plants, participants in this project will learn how to uncover historic collections made by early botanical explorers, document species occurrences across time and space, and then as a result, generate an abundance of data about the plants of our world that can help scientists understand and conserve biodiversity. So much like a library contains books, the herbarium at the New York Botanical Garden serves as kind of a library of plants, but not living plants as you'd see on a visit to our garden grounds. Instead, the collections housed within are dried, pressed, and preserved in a manner which allows them to last for centuries. Over 7 million specimens are housed this way within the herbarium at NYBG, making it the largest collection of its kind in the Western Hemisphere and probably the second largest in the world. Inside are examples of over 200,000 different species, from enormous rainforest trees to tiny wildflowers. And even today, our collection is rapidly expanding with tens of thousands of new specimens added every year from every corner of the globe. So most herbarium specimens are collected by professional scientists on field expeditions, although not entirely. The act of doing this provides them a means to document the organisms they encounter while exploring these wild spaces. Along with each sample, the scientists will often record detailed field notes about where and when each collection was made. Each precious physical specimen, when combined with these field notes, preserves a permanent and unique record of the survival of one particular species at a particular place and time. And in this way, herbarium specimens can be envisioned kind of like the footprints of biodiversity. Over time, accumulation of these voucher specimens by generations of botanical explorers, some predating the founding of the New York Botanical Garden itself, have resulted in an unprecedented record of plant life over the last 200 years. But what can scientists learn today from specimens that have been collected a long time ago? Well, a really important usage of herbarium specimens is monitoring environmental change. So since specimen records go back many years, they can provide unique historic baseline data against which we can compare observations from the present day. For example, in the study of trout lilies, a large data set of observations that included many herbarium specimens indicated a pretty steady shift towards flowering earlier in the season. So this change in phenology, or the timing of flowering and fruiting, seems in apparent response to increasing spring temperatures, which are consistent, unfortunately, with patterns that we see in global climate change all over. So plant specimens are also really important for conservation work. So by mapping where different plant species grow, we can learn about the habitats that sustain them. We can count the number of species that are found in different areas, and then measure the overall biodiversity. Combining species distribution maps with environmental data, we can also predict other suitable places for plants to survive, both in the present day and then looking into the future. All of these tools can allow scientists and land managers to establish priorities and make smarter choices when it comes to conserving natural habitat. Herbarium specimens and the information they contain about plants can also be directly beneficial for humans. So from preserving indigenous knowledge to improving agricultural and forestry practices and providing new sources of foods and medicine. Despite their value for scientists, limited access to collections often constrains their potential impact for research. So even under normal circumstances, traveling to New York City to look at dried plant samples can be very time-consuming and expensive. And in the bizarre world we're living in today, with travel and shipping restrictions in place everywhere, it's now impossible for researchers to visit collections like ourselves and make observations. So how can we then make natural history collections like ours more accessible? So fortunately, there's digitization and citizen science to the rescue. So for the past several decades, and supported by numerous grants from the National Science Foundation, the New York Botanical Garden has been leading the charge to document unique and historic natural history collections electronically. These projects have all involved collaborations between many institutions to digitize particular collections for common research missions. So the goal of each of these has been to capture valuable aspects of collections in our herbaria and natural history museums, and then share them openly through platforms where their data can be used, like the Global Biodiversity Information Facility and our own virtual herbarium. Critical step in all these projects has been just to take a photograph of every specimen that we have. So not every aspect of a collection can be captured in a photo, such as microscopic features of the plant and the structure of the plant's DNA. However, a great deal of useful information can be seen, including the plant's physical appearance and information about the origin of the plant, which is recorded on the collection label. However, with all these photographs we've been producing, there's a tremendous bulk of specimens that still need to be cataloged. And electronic systems are not very good at reading information from collection labels, especially when those labels are handwritten or very old, like many of ours are. But scientists need complete historic records in order to finish their work, which means information like where, when, and by whom each specimen was originally found, we need to collect still. So this is where citizen scientists come in to help professionals collect essential data from herbarium specimens. The key platform we use for this work is called Notes from Nature. The projects are online, so all you need is a computer with internet access to participate. Your mission is to view photographs of plant specimens, decipher their typed and handwritten labels, and then answer questions about the geographic and historical origin of each one. So Notes from Nature is a collaborative crowdsourcing project in which many institutions can lead expeditions like this to help uncover scientific data. So from the main page, you can navigate to the New York Botanical Garden project page by clicking our icon with the NYBG logo. And from here, you can embark on virtual expeditions to recover data from our herbarium. Each one targets a specific set of specimens to accomplish unique research mission. These may include projects dedicated to exploring particular geographic regions, or investigating evolutionary relationships of plant groups, or documenting the journeys of notable early explorers. Right now, we have 2 expeditions online, one featuring orchids of the United States. And for this, we're asking folks to record all the information about where, when, and by whom each plant was collected. For our other active project, which is called NYBG Globe Spotter, we're looking at specimens coming from all over the world, which have been added relatively recently to our herbarium. For this, we're just asking for the basic geography and what country was the specimen found, and if it's in the United States, which US state, making it quite a bit easier and a good introduction to reading natural history specimens. So when you open an expedition for the first time, you'll see a tutorial pop up. There are many resources in here to help you get started, including a basic walkthrough, links to a training video, and other tips you'll need to interpret specimens and enter information. When you're ready to begin, you'll see the specimen on the left and navigation tools, and then the fields you'll be entering information in on the right. For each specimen, you'll need to locate the collection label. So this describes the origin of the plant, and then you'll record all the details by filling in those relative fields on the right side. If you need any help understanding these tasks, there's a help button always available, and you can click next to proceed through multiple pages to advance to all the questions. In the end, you'll click done and then submit your observation, and then researchers have had access to that data. Alternatively, you can click done and talk and share a comment along with your classification. This is a really fantastic tool because through it, you can reach our online community. All comments about NYBG specimens are posted automatically to our community talk forum. And this is a space where participants can ask questions, receive and provide feedback to each other, share research sources and interesting discoveries, and then engage in the collaborative effort together to solve challenging historical puzzles. Sometimes we have as many as 100 comments per day, so this is a very active forum for communication in our project. Since the fall of last year, we've had over 200 users participate, and 600 users participate, and we've completed more than 130,000 classifications, which is pretty substantial. From our main page, you can navigate to your Notes from Nature Garden Fieldbook once you've signed in and registered for an account, and you can track your contributions to the project, including your overall total, the daily and weekly averages. Recent specimens you classified will be archived here, and then you can acquire badges and things as you reach important milestones. So every specimen you classify and the data you collect is published freely for everyone on the New York Botanical Garden CV Star Virtual Herbarium and other data aggregation portals for natural history specimens. Here, the date, the location, the person who collected the plant, it's all easily searchable and accessible for scientists around the world. And all those data points, when combined together, helps us reveal that bigger picture about nature and where plants are found in our environment. So why participate in these virtual herbarium expeditions? Well, you can make a real difference by helping botanists around the world build these new digital data sets. They contribute to our understanding of the living planet, and they open new avenues to investigate long-term biodiversity change that were never accessible before. You can also challenge yourself and explore natural history collections and do your own research to solve historical puzzles. Who was this mysterious botanist, and where were they collecting orchids in the year 1850? Finding that answer and learning more about these explorers along the way can be an incredibly rewarding aspect of the work. So finally, you can find community along with the many other citizen scientists in our project that are working towards this shared mission of discovery. And although this is kind of what community looks like these days, with all of you meeting through virtual spaces, citizen science projects like Notes from Nature that allow us to work remotely and share together these experiences can be really helpful to stay active and stay connected. So if you're interested to learn more, I encourage you to join our follow-up breakout session today at 3 p.m. We'll discuss keys to interpreting herbarium specimens along with strategies for navigating challenging labels. You'll learn about tools and resources you'll need along the way, including how to use the New York Botanical Garden's own virtual herbarium and advanced features of the Notes from Nature portal. We'll also do live and interactive demonstrations together, so all of this makes more sense. And I'll invite all of our attendees to participate in those as well. You're also welcome to get started on your own. So go to notesfromnature.org, click on the icon for NYBG, and you can join our virtual expeditions that way. We've got resources available there to help you get started, and so you can have fun. If you'd like to learn more about the collections in our herbarium and how they're used for science and the unique and sometimes whimsical stories they tell about the natural world, you can discover them by visiting the Herbarium Hand Lens at the website shown here. So that's it. Thank you so much for listening and for all your help and from our amazing participants without whom none of this would be possible. If there are any questions from the audience, I'm happy to take them later on during the breakout session.
Great, thank you so much. I learned a lot, and I always get really excited about Notes from Nature and all our virtual opportunities. So that was so fabulous. All right, now we have Esther. So Charles, if you want to, all right, great. Now Esther, you can share your screen, and you're our last presentation before the breakouts.
Excellent, thank you. Thanks so much, Charles, Christine, Daniel, and Carolyn. This has been so great so far. I've learned so much. All right, I'm going to share my screen. Okay. All right, perfect. Okay, so hello again. My name is Esther Jackson. I'm the Public Services Librarian in the Leicester T. Mertz Library here at the New York Botanical Garden, and I'm also the Wikimedian in residence. So today I'm going to talk a little bit about the citizen science or participatory science projects that we have undertaken in the library using the library collections with the help of citizen scientists here at the garden. And there was a question earlier in the chat about citizen humanities and whether NYBG does anything in that avenue. Because the collections of the library are interdisciplinary and there is so much to do with the history of science, the wiki projects that we've taken part in could also be viewed through the lens of citizen humanities for those who are interested in that. So just a quick introduction to the collections at the library. We're the largest library of our kind in the world. So we collect in the fields of botany, horticulture, and landscape design. We have over 500,000 volumes in our library between books and journals and other bound materials. And we have millions of archival collection items, from correspondence to manuscripts, and a lot of art collections as well. So from botanical watercolors to photographs and lots of other things. There's a huge collection. And so like the herbarium, we often have to ask ourselves, how can we make these materials as accessible as possible to as many people as possible? And to that end, the library has also been doing digitization work for many years to get our collections out and available to as many people as possible. And we do that in part through efforts by citizen scientists using our collections to get materials into things like Wikipedia, Wikimedia Commons, and Wikidata. So I'm just going to take a moment to show the library homepage. So you can access the library homepage from home. With the search our catalog box, you're able to find materials that you would be able to view in the library in person, as well as things that you'd be able to look at digitally if you were coming to our collections remotely. If you're specifically interested in digital collections, as we are currently closed to the public because of COVID-19, you can click on digital collections, and this will give you a list of different libraries and collections where our materials are shared. I'm just going to mention two really quickly. The first is Mertz Digital, which includes digitized materials just from the Leicester T. Mertz Library. So we have things related to the New York Botanical Garden specifically, such as historic photos. We also have journals, seed and nursery catalogs, and books. And then the Biodiversity Heritage Library is a digital collection that includes materials from libraries all over the world related to biodiversity. And that includes scientific works, it includes scientific illustrations, and it's fully accessible from home. And I like to show off the Flickr photo stream because you can see some of the materials that are accessible to people working remotely. Okay. So I mentioned Wikipedia. Wikipedia is a fantastic site through which we've been able to make some of our content more available, and it's a really exciting and huge participatory science resource. It is the largest and most popular general reference work on the World Wide Web, and anybody can edit it, anybody can add to it. So you can work on content related to the garden, you can work on content related to botany, and you can work on content related to other topics. So it's very interactive, very participatory. There are 5 pillars of Wikipedia, and the one that I wanted to highlight for the talk today is the fact that the content in Wikipedia is free to use, free to edit, free to distribute. And so it's a very powerful resource for science and for the humanities because so much of the content, because all of the content is available. And that's really, I think, part of the ethos of citizen science projects in general, this idea of openness of data, this idea of openness of information that can be used by so many different people for so many different projects. And so again, what we're trying to do here in the library, what we have been doing is get materials from within our collection out into the world. And so we've done that through Wikipedia, which I just mentioned. So editing and creating articles about scientists and botanical illustrators. We've contributed content to the Wikimedia Commons, which is the media repository for wiki projects. If you see a picture in a Wikipedia article, if you see a photograph or an illustration, most of the time that's coming from Wikimedia Commons. So that's an incredible resource of materials that are in the public domain. So things that were produced or published before 1925 in the United States. And then also works that people have released, that they have created and released into the public domain. The final project that we've contributed to is Wikidata, which is the structured data component of the Wikiverse. And so that's really popular with librarians, with archivists, with scientists who have data that they would like to share that is in a more structured format. So a Wikipedia article would be more of a narrative form, and then Wikidata would be a way for you to represent information about people, about objects, about plant species in a more structured fashion. So also very useful. So one of the ways that we've gotten our content into Wikipedia through the help of citizen scientists and citizen humanists is through editathon workshops. And so this is an image from an editathon that we had specifically to share information about female scientists and to take information from our collections and information in the Biodiversity Heritage Library and put it into Wikipedia in a way that more people could access it and interact with it. And so we focused on female scientists and in other events, female artists, because of the gender gap in English language Wikipedia. As of January 2020, less than 20% of the biographical articles in Wikipedia were about women. And there's lots of reasons for that being the case. And thankfully, we have materials in our collections that help us to close that gender gap and help represent and make available the work of female scientists and female artists. The other campaign that we took part in to do this work was the Her Natural History campaign, which was through the Smithsonian and the Biodiversity Heritage Library. So this campaign that you can find information about online was a way by which libraries that were part of BHL, so libraries with large natural history collections, were able to highlight the work of important scientists and artists within their collections. So we had a couple of events where people were able to make edits to Wikipedia, Wikidata, and Wikimedia Commons. And we also had a couple of Facebook Live events where we showed off some of the materials in our collection with staff from the herbarium and the laboratory. And again, we were able to make this content more accessible through the work of citizen scientists who were instrumental in those events. So I'd like to take a moment to show a minute of a video. And the interesting thing about Zoom is that audio doesn't work super well. So what I'm gonna do is just play 1 minute of this video, and there are captions associated with it. So I'm gonna play it and let people read it, and then I'm gonna pause it and come back to the presentation. So as a little bit of context, this is a video from an editathon that we had in the library, and you're going to hear from 3 people who were at that event, and they're going to talk about the work that they did. It's a common thing. So translating content from one language Wikipedia into another language Wikipedia is a really important thing that people can do. So translating from Russian to English, English to Russian. So filling in gaps, very important. All right. So the last participant was talking about using digital resources in order to enhance Wikipedia articles. So she was working with JSTOR and looking at papers that Anna Marie Vale had authored and then representing that information in Wikipedia. All right. So much clicking. I also want to mention and highlight a specific project in the Wikimedia Commons. So I talked about editing Wikimedia Commons and contributing there is another really important thing. And so this is the Wikimedia Commons page for content from the Biodiversity Heritage Library. And so this project is run by a volunteer named Siobhan Leachman, and she's put together just a lot of resources for people who are interested in editing in the Commons, people who maybe want a more visual experience as opposed to a textual experience. And so there's resources here, and there's also task lists for things that people can focus on editing. So that's a really wonderful thing about wiki projects. the way in which people from around the world come together to create lists of work that they think need to be included in the Wikiverse. And so there's a lot of points of entry to these projects. So the breakout session that I'm going to lead after this has a number of objectives, but it is kind of dependent on who decides to come to the session. So there's going to be a couple of questions, poll questions, before this part of the webinar ends. If you could answer them, that will help me have direction for the next hour. If you're going to be coming to the library breakout session, this wiki session, it's helpful for you to have a desktop computer so that you can follow along, but you're also welcome to come and observe and ask questions and, you know, just generally nerd out about information. That's what we're all here to do. And if you have any questions after this event, please feel free to contact me. I'm putting my email up on the screen there. I'm always happy to answer questions. And if you are local and you're looking for information about other New York City opportunities or programs, the Wikimedia NYC group supports the work at NYBG and also supports the local community. And that's a very great group as well. Thank you so much.
All right. So if you want to stop sharing your screen, all right, I'll share mine. 1 second. We're gonna have 2 quick polls before I explain how breakout sessions work, and everyone can vote in these 2 polls. Just out of curiosity, we just love to hear a little bit about your answers to these. So we want to know, after you heard Esther's awesome presentation, have you ever edited Wikipedia before? So I'm launching the poll now. Please feel free to vote. Oh my gosh, we got about 80% no's so far. Keep on voting, keep those votes coming. All right, last chance to vote. 3, 2, 1, and we're gonna end the poll. So the vast majority of people attending have not edited Wikipedia before. And then we'll do one more quick poll for the whole audience. We want to know, do you already have a Wikipedia account? All right, so we have launched the poll, so please feel free to vote. Got a lot of no's coming in, a few not sure's, and 2 yeses, which is kind of cool. 2 people know for sure they have one. All right, last chance to vote. 3, 2, 1, last chance, and we're gonna end the poll. So yeah, about 70% no's, 2 yeses, and 26% not sure's. Awesome. Well, thank you so much to all of our speakers today.